Let's start with immunology, and the questions I have totally collected questions that have been asked since 10 years, all in the DNB still. First question goes like this: in utero infection leads to which immunoglobulin? Yes? Because we do not have a chatting board, I don't know, I am able to interact with the online people, but fine. IgM, all go with IgM, or shall you have any other answers? In utero infection leads to which immunoglobulin? Yes, it's IgM, any infection, because the fetus starts generating when it's in the gestation period, 23 weeks of fetus life only, the fetus starts generating IgM. So it's not IgG, because IgG could be a false or an anamnestic reaction. It will produce an anamnestic reaction because the maternal IgG antibodies may transfer through placenta. So always it's the IgM, and that's what you have to look out as a doctor also tomorrow when you are suspecting any congenital infections in a fetus. Am I clear about this? So the answer is IgM. Immunoglobulin that is inactive at high temperature. The beauty of IgE is it's all exception. If you just go back, look at Ananthanara 9th edition, if you see, look at the table, IgE has lot of exceptions. Like it's the only immunoglobulin which is low molecular weight, it's the only immunoglobulin which is heat labile, it's the only immunoglobulin that participates in the allergic and parasitic infections. It is present as a receptor on the mast cells, it has receptors on the mast cells, basophils. Okay? So these are exceptions. All immunoglobulins are heat stable except IgE. So the answer is IgE. Inactive at high temperature, it gets destroyed. Immunoglobulin present in local secretions, IgA. So that's, you can just feel that it's very basic questions that is asking in immunology. Only thing you have to know wherever, because that's the reason why it's been given the name secretory immunoglobulin. IgA is the answer. Secretory IgA, the name itself, that's wherever there are secretions in your body, right from your mouth to anus, it is basically the respiratory tract, genital urinary tract and gastrointestinal tract. It is where this IgA immunoglobulin protects against the local, it provides also local immunity, it also participates in innate immunity. Am I clear about this? IgA immunoglobulin participates in innate immunity and also your local immunity. Immunoglobulin which crosses placenta. Very simple, God's gift, remember like that, gift from mother to the child. So it's IgG that transfers from the mother to child. Which immunoglobulin transfers through placenta? IgA. Which immunoglobulin passes through colostrum? IgA. Which immunoglobulin is present in Peyer's patches? Very simple, Peyer's patches are something in the small intestine again a GI tract, GI is again a IgA. Which immunoglobulin acts as a receptor on B cell? Which immunoglobulin acts as a receptor on B cell? B cell by default, if you know pre-B cell, pro-B cell, when the B cell is being formed in the bone marrow, unlike as T cells in the thymus, the B cells by default have receptors for IgM and also IgD. Okay, so it's the IgM. So this is your B cell and this is your immunoglobulins which are act as receptors by default. And this is the marker. How do you identify B cell is CD19, 20, 21. There are some markers. How do you identify a B cell? It is CD19, 20 or any B cell that is a marker as, or any cell. You can say any cell that has a marker as CD19, CD20, 21, it is a B cell. Any cell that has CD3, it is a T cell. It is a universal marker for T cell. What is the marker for natural killer cells? 16 and 56. 16 and 56. So these things are very important that you have to remember. They are very high yield in examination. Pentameric antibody which has a J chain, IgM. IgM is a pentamer, M millionaire molecule, highest molecular weight, highest intravascular distribution, sedimentation coefficient IS. Write down the mnemonic MIS. For IgM, the mnemonic how to remember IgM, important exam point of view is MIS. M for molecular weight because it's a pentamer. Valency is 10. Raju, if there is a problem in the online, let me know. Those people who are not satisfied or they are okay also. If I am going fast or something like that. IgM is a pentamer. The mnemonic for IgM more extra. MIS. M for molecular weight, highest. I for intravascular distribution. S for sedimentation coefficient. That is also highest. This is for IgM. Write down IgG. The mnemonic to remember IgG exam point of view is DHS. D for daily production, H for half-life, S for serum concentration. We will be dealing with exam questions. Simultaneously, I will be teaching you more than that what can come as a question. These are all the previous, because what can come is what I am going to teach you simultaneously. Which is the other immunoglobulin which has J chain other than IgM? A, because a dimer. J chain is present in IgA, which is a dimer, Pentameric IgM. So these are IgM molecule with a, pentamer with a J chain and all. Action of papain on an IgG molecule produces what? Suppose this is your FAB portion. Do you remember this? And this is your FC portion. This is a typical immunoglobulin. This is an FAB. FAB is antigen binding. Antigen binds at this. This is hyper variable region, because you don't know how the antigen is going to come. It is a big antigen, small antigen, different shape, arrangement. So it keeps on varying. It has to vary its size. Hyper variable region, and this is a constant, doesn't change at all. FAB antigen binding. FC is the complement binding. And you can remember another way also. You can remember this is an amino terminal. AMINO. This is an amino terminal, whether this is carboxy terminal. You can remember C like that also. The question is, action of papain on IgG molecule produces what? Papain acts at the hinge region. This is the hinge. This is a heavy chain, this is a light chain. I think you know about this. This is a heavy chain because it is heavy, and this is the light chain. Papain acts as the hinge region. So it produces 2 FAB portions with 1 FC portion. Okay? Whereas if I ask you one more question, pepsin acts and produces what? It produces something like this, right? 2 FAB portions and 2 FC portions, right? Pepsin acts like this, whereas papain acts like this. So that's what the figure shows. Secretions are rich in which immunoglobulin? Wherever these word secretions are coming in immunoglobulin, no doubt, it's IgA. Classification of immunoglobulins is based on heavy chains, basically. Classification of immunoglobulins is based on the heavy chains, actually mu, gamma, delta, epsilon, okay. That's the reason why we get G, then alpha, A. This is mu for M, G for A for alpha, gamma for G, delta for D, epsilon for E. Based on the nature of heavy chain, the immunoglobulin is classified into 5 types, whereas light chains are similar in most of them, that is kappa or lambda. Kappa or lambda. But light chains play a very important role, medical point of view. What do they cause? What is the light chain disease? Multiple myeloma. Ben-Jones proteins are secreted, those things you have to check. Or beta macroglobinemia is another disease, the light chain diseases. So basically there are 5 types: IgM, IgG, IgD, IgE. IgG, D and E structures are same, as you can see, but M is a pentamer, where IgA is a dimer. You have a monomer IgA also. Monomer IgA is also there, but it is serum IgA. Dimer IgA is a secretory IgA. And anything 2 or more than 2, you have a J chain. Okay? Here also you have a J chain. Pronitz-Kutzner reaction demonstrate. Basically, it's a case of passive cutaneous anaphylaxis. Passive cutaneous anaphylaxis, and it is mediated by IgE. It is mediated by IgE. There are two people, Prusners and Kussners. The two people experimented on themselves. Prusners was actually allergic to fish. He was allergic to fish. So let's look what they have done actually. So Prusner reaction they have demonstrated passive cutaneous anaphylaxis. The answer is IgE. They detected atopy antibody. Serum collected from Kussner, who had an GI allergy to certain cooked fish. Kussner has an allergy to a fish. So when he had taken ingested, he had a reaction. And serum was collected from this Kussner and was injected intracutaneously into Prusners. Followed 2 hours later by injecting intracutaneous small amount of cooked fish. So suppose I have allergy to a fish, I have eaten it, they have collected my serum and injected into you. Suddenly they take the fish extract and also inject it into you. So when serum is injected into you, antibodies would have developed into your body. Now this fish extract which has given into you, that are antigen specific. Same fish, that fish extract is being given. I am allergic to that fish, my antibodies serum is being given. Into that antibodies will develop and this antigen they will cross react and a wheel and flare reaction will occur. And that is because of IgE, because mast cells, basophils will get stimulated in your body. Followed 2 hours later by injecting intracutaneous small amount of cooked fish extract into the same site, a wheel and flare reaction occurred. This specific affinity of IgA, that is called reagenic antibody for skin cells, forms the basis of PK reaction. Now why this? It occurs only in humans. You cannot demonstrate this reaction in other people because IgE is homocytotropic, not heterocytotropic. It is homocytotropic. Only in Homo sapiens or Homo people you can, human beings only you can. It is species barrier. Test is not done these days because of risk of transmission of hepatitis, because you inject, take needles, all this, so they have stopped because there are so many other reactions where you can demonstrate, like ELISA or radio allergo sorbent test, okay, passive agglutination test. The other test where you in vitro in lab you can demonstrate. What are the serum levels of IgG? I gave you a mnemonic, right? IgG is what? DHS. So daily production is high, half-life is high, serum concentration is high. What are the serum levels of IgG in milligrams per milliliter? So it's basically serum concentration 10 to 15. Actually its answer is 12. 12. Serum concentration maximum is 12. True about IgM. Most heat stable compared to IgG, activates complement, is not synthesized by fetus, crosses placenta. Let's go by the negativity rule first. Does it cross placenta? IgM? No. Is it synthesized by fetus? True. Is not synthesized by fetus. Activates complement? Yes, it might activate but not specific. Classical complement pathway or alternate pathway or Mannose binding Latin pathway not mentioned. Most heat stable compared to IgG? No. The most heat stable immunoglobulin is IgG. IgG is most heat stable compared with IgM and IgG. The more appropriate, yes, this can also be the answer. You can defend again, fight with me, but most appropriate answer is A, most heat stable, because IgM can also do, IgG can also do, but nothing like. Then there's an exception, there is rule taken out, then the answer would be IgG, because IgM is not heat stable compared to IgG. Here, one more thing I would like to comment coming about immunoglobulins. Another question that can ask in AIMS basically is which immunoglobulins are warm antibodies? Warm antibodies, cold antibodies. IgG is warm antibodies, IgM participates in the cold antibodies, especially the cold agglutination test done for Mycoplasma pneumoniae. Cold agglutination, which you are going to study like pre-lunch or post-lunch. Mycoplasma pneumoniae, you do a cold agglutinin test. For there you have to use a IgM antibodies. How to remember that? IgG means garam. Garam in Urdu means hot. Garam, remember like that. Here that's how to remember this also, because heat stable again. More the heat it is stable, IgG. So two things can be covered, concept and also for the exam point of view. G means garam, IgG warm antibodies, and also G is heat stable. That means to say is M is cold antibodies. First immunoglobulin to appear following infection, no doubt, is IgM again. First immunoglobulin to appear anywhere, first infection. Commonest immunoglobulin deficiency is, should be well versed with the immunodeficiency disorders is a topic in Anantanaran in microbiology they teach. Primary immunodeficiency, secondary immunodeficiencies and all. The most common immunodeficiency disorder, which immunoglobulin is IgA. Can you give an example for IgA? Common variable immunodeficiency IgA causes what type of infections? Mucocutaneous candidiasis is caused by T cell. No doubt, very good, well attempted. Common variable deficiency caused by IgA causes Giardia infections. Giardia. Which antibodies is most commonly produced in secondary immune response? IgG. Secondary immune response, past infection, because earlier you had an infection, so your body has produced antibodies, and once the antibodies are there in your body, always they will be in your body, like hepatitis, dengue. You clear? You got my point? So once you have an infection, antibodies will be produced and they will be there in your body till lifetime. Though you don't have infection also, you do a dengue test, it will come positive only. Don't get worried, because your body has already produced. But if you have an infection, your body already knows, okay, I already have it, then it will start fighting very fast. That is the safety of antibodies being produced in your body. That is because of clinical supplementary infection, natural acquired immunity you get. So which produces secondary immune response? IgG. Which immunoglobulin does not activate complement or does not participate in complement pathway? E. There is no role of E at all. It is G and M which participates in the classical complement pathway. It is IgA that participates in the alternate pathway. The mnemonic to remember that is GM, which is a company that makes the world's best cars in the world. There is a company that makes world's best cars, GM Motors, General Motors. It's the name of a company. GMC. Have you seen movies Fast and Furious, Part 6, Part 7? The big vehicle, the hero rise. It's GMC. Most of the foreign countries and all in Hollywood movies, if you see, it's written as GMC, General Motors Corporation. General Motors makes classic cars. So G and M participates in the classical complement pathway. IgG and IgM participate in the classical complement pathway. It's very easy to remember IgA because IgA is A and alternate pathway also starts with A. In IgG, again, if you do an isotype switching, something called isotype switching because of the variable VD-J chain, variable dominant and the J chain, because of the reassortment in the genes in the VD-J chain, isotype switching of the immunoglobulin occurs in which IgG1, G2, G3, G4. G1 and G3 participate most quickly with the complement that is released first, QRS, C1Q, whereas G4 do not react, less reactive to G2. Okay, this is one extra thing again in AIIMS, PGH, Chandigarh you might be asked, but not in DNB. Just for your knowledge that not all IgG, when it's talking about isotype switching, IG1, IG2, IG3, IG4, which isotype of IgG does not participate in complement means the answer would be IgG4 does not participate in the pathway. Which of the following antibodies shows anamnestic response? Again it's IgG. Anamnestic response is you don't have an infection but antibodies are there in your body. You don't have an infection but antibodies, when you do a test, antibodies are there in your body. Similarly like typhoid, cross illness, cross reaction, you have illness because of other bacterial infections also and you suspect it's typhoid but it's not typhoid. Anamnestic reaction is IgG. Which complement binds with Fc portion of IgM in classical pathway? Yes? C1, no doubt about it. Which complement binds with the Fc portion of IgM in classical pathway? Just to touch with the complement pathway, because there is no question paper without the complement in DNBs. This is the liver, and liver starts whenever there is a bacteria, antigens are present on the bacteria, you agree with me? And whenever antigens are there, they stimulate the body, lot of things occurs, T cell, B cell and all, finally B cell can be plasma cell is immunoglobulin, and this is the FAB site as I told you, and this FC portion which is free, FC portion which is free, it stimulates the liver and start releasing the complements. C1, C4, C2, C3 in this order, all A's and B's are released. Okay, A is eaten away, A for apple, remember A, B for baby, baby eats the apple, progresses in life, B stays behind, and again C1 also, C1, Q, R, S, don't go into details because the computer exam, it's not a theory answer, but you have to remember this order, 1, 4, 2, 3, and basically it's a C3B, and C3 convertase. It acts on C3b and causes C5, C6, C7, C8, C9 release in this order, which is called membrane attacking complex or terminal complement pathway, which acts on this bacteria and bring pores. That's how the bacteria is killed. Okay. Now what you have to remember, a lot of things over here is, this is classical complement pathway. Coming to the alternate pathway, it is, there is no C1 here, directly C4. Suppose this is the liver, C4, C2 and C3. So what is the difference between alternate and complement pathway? What is missing? C1 is missing. Mannose binding pathway, mannose binding lectin pathway. It's nothing but this is the bacteria, in this mannose sugar is present and the lectin from the endothelium will bind to this. Mannose binding lectin pathway, and it starts with directly C3. So what is missing in C1 is missing, C4 is missing, C2 is also missing, 1, 4, 2, everything is missing here, directly starting with the C3b. So if I ask you a question now, what is the starting point before the membrane attacking complex, or what is the common pathway? C3, B should be the answer. Whatever from wherever they are coming, the common source or the pathway is C3b is an exam question. Now there are 3, 4 other things you have to remember. Here when I am talking about C3a, C3b, C5b's, again here also A is written, 5b is written. There are 3 things you have to remember: C3a, C5a, C3b, C5b. Because all those things that are gone are not waste, some are very important. Like C35 is a chemoattractant, or also they are called anaphylatoxin. C3b and C5b acts like opsonins. They act like opsonin, but the best would be the C3a and the best here would be C5b. C3a, C5a, they act like chemoattractants, they attract more neutrophils. They tell or sense the body that there is a need for neutrophils to pour in here so that we help the tissue to not get much destroyed or inflamed. So they attract more neutrophils, and C3b, C5b are called opsonins. There are other also like leukotriene B4 and other interleukins, which we are going to talk about that in a few minutes from here. So there are some extra things that you are remembering on the complement pathway. What is the membrane attacking complex consist of? 5, 6, 7, 8, 9. It's a complex, and there are a lot of questions I think which we are going to study now. Right, that's C1. Next question. That's what I explained you. C1 complex, convertase, C3b cleaves, this is the C3b, cleaves into all this 5, 6, 7, 8, 9 and finally destroyed this. Classical alternate starts with, okay, that's it.